Hello and welcome back to Immunology The War Is Over, episode 6, where we'll be diving into all things B cells and plasma cells. In the last episode, we got to know in great detail the magical ability of antibodies, but antibodies wouldn't even exist without the input of B cells and plasma cells. And so in this episode, we're going to find out exactly what happens when B cells become activated, including how T cells help in this process. Okay. So B cells, after being awarded their IgM and IgD B cell receptor, leave the bone marrow and hang out in our secondary lymphoid organs such as our spleen, lymph nodes and Peyer's patches in the gut. And inside those lymphoid tissues, B cells hang out in particular areas known as follicles. And follicles are so much fun. When B cells are inactive, you'll find them around the edge of the follicle. But when they're activated, they will migrate into the middle, forming what's known as a germinal centre. And germinal centres are where the magic happens. And this setup reminds me of family gatherings. You know when someone has a wedding and they hire a function room and there are tables and chairs around the edge of the room and then the dance floor is right there in the middle? Well, this is exactly what is happening inside a B cell follicle. Around the periphery, we have B cells chilling out at their table, waiting for their respective antigens or dance partners to come along. But when they do find their antigen match, they'll head straight to the dance floor, the germinal centre. And remember, this is not the only party happening inside this lymph node. Right next door to the B cell dance party is the cuddle party that we saw in episode 2 between T cells and dendritic cells. So lymph nodes are where the party is at. Next time you have an infection, I want you to feel that big swollen lymph node in your neck and remember all the fun stuff that's going on in there. Cuddle parties, dance parties, you name it. Anyway, back to the follicles. This is where naive B cells gather to wait for antigens. And antigens are constantly being drained into lymph nodes from surrounding tissues. So let's take a look at what happens when a B cell comes across its matching antigen. Antigens within tissues hitch a ride on extracellular fluid and drain into a nearby lymph node. There, they will encounter the follicle. Mother Nature has strategically placed follicles in such a way that antigen encounter is highly likely. Mother Nature also made sure that the B cells within follicles are constantly turning over, travelling around to different follicles and different lymph nodes around the body, thereby increasing the chances of a B cell finding its matching antigen. When an antigen binds to a matching B cell receptor, the B cell will internalise that antigen, process it, stack it onto MHC2 molecules, and show it to nearby T cells. And B cells do this for a couple of reasons. Firstly, because they can. B cells are antigen-presenting cells, and they show antigens to T cells just like dendritic cells and macrophages do. But B cells also have another motive in mind. They actually need guidance and encouragement from T cells in order to know which antibody class to produce and in order to differentiate into plasma cells and memory B cells. So they need T cell help. And the good news is that their T helper buddies are never far away. Right there on the edge of the follicle, the B cells are literally surrounded by T cells. So the B cell goes to the T cell, presents the antigen, and sparks off signal 1, 2, and 3, which we covered back in episode 2. The T helper cell takes this signal, as well as the wider cytokine signalling around the place, and differentiates into various subsets depending on what is needed for that particular pathogen. And as we saw in episode 2, T helper subsets are defined by the cytokines they produce. And the cytokines they produce will help the B cell to know which antibody class they should be generating. So for example, Th1 cytokines promote IgG antibodies, whilst Th2 cytokines promote IgE or IgA antibodies. And so cytokines generated by T helper subsets are important for B cells to know what to do. And in addition to this, there is also a particular T helper subset which is dedicated to helping B cells to do their thing. And these T cells are known as the T helper follicular helpers. These little guys make it their mission to support B cells in becoming the plasma cells that they were destined to be. And they also make sure that they generate the highest affinity antibodies that they possibly can to a particular antigen. Most of these T helper follicular helpers position themselves around the edge of the follicle, right next to the B cell. But a few cells gain access to the germinal centre, where all the B cell action happens. So after a T cell is recruited, activated B cells head to the germinal centre for a shape-shifting dance party known as affinity maturation. This B cell is about to grow up real fast and be forever changed. And this is fascinating. Affinity maturation is all about encouraging this B cell to make the best antibody it could possibly make against the antigen in question. This is different from class switching. We said before that cytokines from T cells will guide antibody class switching, but remember that the class of the antibody is determined by the heavy chain. And class switching only changes the heavy chain in such a way that it doesn't affect the variable region which binds the antigen. So class switching doesn't affect the antigen binding site. But there is a separate process which will now occur in order to make sure that antigen binding is as tight as it possibly can be. And this process is known as affinity maturation. Let's unpack that now. We saw in the last episode how developing B cells in the bone marrow shuffle their DNA in order to create their unique B cell receptors IgD and IgM with a unique binding site against a particular antigen. This DNA combination was locked in at that time, and so the B cell continues to generate those same B cell receptors from then on in. However, once a B cell comes across its antigen and recruits the help of T cells, that unique binding site that was previously locked in will now be modified in order to create a better binding site with the best possible fit for a particular antigen. The immune system really is incredible, but it's also alarmingly reckless. In order to make this better antibody that binds even more tightly to the antigen, the B cells must engage in a process of trial and error, and they do so using genetic mutations. I'm not kidding. B cells literally allow their DNA to be mutated, not shuffled, mutated, which, let's be honest, sounds rather frightening and makes you wonder why you don't develop lymphoma every time you get a cold. But Mother Nature thought about this. She actually made sure that B cells are very prone to suicide. So unless B cells get a survival signal from their T cell buddies, they'll self-destruct. Thank you, Mother Nature. I am grateful for this safety feature. And this survival signal from T cells takes the form of cytokines, especially interleukin-4. Interleukin-4 is B cell survival fuel, and this is produced abundantly by the T helper follicular helpers. So in the germinal centre, we have multiple clones of that original B cell which bound to the antigen. And each B cell clone is randomly mutating its DNA using specialised enzymes. So each of these clones will be producing a different variable region from each other. But only one B cell with the highest affinity antibody will be selected from the group. So the B cell clones are competing with each other to make the best possible antibody for this antigen. And helping to judge this competition are follicular dendritic cells. These are specialised dendritic cells which have only one role, helping B cells to make those high affinity antibodies. So the follicular dendritic cell holds out the antigen on their projections, and the B cells get to try out their updated antibodies on these antigens. So that's affinity maturation. Once the highest affinity antibody is realised, the B cell producing the antibody will be selected to proliferate into a colony of B cells, and this is known as affinity selection. This colony of selected B cells will now take one of two paths. Many will differentiate into plasma cells, which are essentially antibody factories. Plasma cells relocate to the bone marrow, and from there they secrete oodles of high affinity antibodies over and over and over again into our circulation for the entire lifespan of that plasma cell. So until that plasma cell dies naturally, it will continue to secrete those antibodies into our circulation. The B cells which don't become plasma cells differentiate into memory B cells, which express those high affinity antibodies on the surface. So, should they encounter that same antigen again in the future, they will respond swiftly and directly, skipping the affinity maturation and selection processes. And what I've described to you so far refers to the journey of a single B cell responding to a single antigen. But in reality, microbes have lots of antigens on their surface for B cells to get excited about. So during an infection with a particular organism, there are going to be lots of B cells recognizing lots of different parts of the microbe, becoming activated and generating high affinity antibodies to all those different parts of the microbe. So, in response to a single organism, we make lots of different high affinity antibodies, and this is known as a polyclonal response. A polyclonal response really just means a normal response to infection. Lots of different B cells making lots of different antibodies, doing their thing. A monoclonal response, on the other hand, would be associated with, say, a plasma cell malignancy, such as myeloma, where the same antibody is being secreted over and over and over again. That tends to be a malignant phenomenon, whereas an infection would provoke a polyclonal response. And it's worth mentioning that this action-packed germinal centre only exists when B cells become activated. So a resting lymph node with no infection, there's not going to be any germinal centres around. But say, for example, you have a vaccination. In the days following the vaccination, you'll develop germinal centres in the lymph nodes draining that area. And these germinal centres will be working to find the best antibody against that antigen that you were vaccinated with. Now, the process we've just described is the pathway which involves help from T cells. But there are some B cells which operate independently of T cells. In fact, there are two main types of B cell, B1 and B2. B2 cells are the ones we've described so far in this episode. They are your typical B cell that you'll find inside the lymph node follicle, and they require help from T cells. B1 cells, on the other hand, hang out in our body cavities, such as the peritoneum and the pleura. There's also a special subset of these B1 cells which are located in the marginal zone of the spleen. B1 cells can directly respond to antigens they encounter. They don't rely on T cells, and they are particularly good at responding to lipids and carbohydrates on the surface of microbes, such as bacterial lipopolysaccharides. And so in response to seeing a microbe, they can directly release antibodies. Now, because they are working independently of T cells, these antibodies will only ever be IgM antibodies, and they're not going to undergo affinity maturation and selection to create the best ever antibody for that pathogen. So it's not fancy, but it's still a handy feature that we have B cells in our body which can directly respond to bacterial pathogens that they come across. And splenic B1 cells deserve a special mention. These hang out in the marginal zone of the spleen, and of course, whilst lymph nodes drain extracellular fluid from tissues, the spleen is scanning our bloodstream for pathogens. And these marginal zone B cells are in prime position to combat bacteria in the bloodstream. And they are particularly good at producing antibodies against structures on the bacterial cell walls. And you may already be familiar with this concept. People who've had a splenectomy are more prone to infections with encapsulated organisms, and this is one of the reasons why. Post-splenectomy, patients are managed with amoxicillin prophylaxis, and they must be vaccinated against encapsulated organisms such as pneumococcus, meningococcus, and Haemophilus influenza. And in the interest of highlighting the talents of B cells, it's also worth pointing out this recognition of carbohydrates and lipids on microbial surfaces is a magic trick which is particular to B cells and something that T cells cannot do. In general, for an antigen to be recognised by a T cell receptor, that antigen has to be packaged into a peptide fragment and shown to T cells on MHC molecules. The B cell receptor, on the other hand, takes the form of an antibody, which means that it can recognise and bind to 3D structures. And those structures might be lipids, carbohydrates, proteins, you name it. So the B cell recognises raw antigens on the surface of microbes, whilst T cells require the antigens to be neatly packaged and delivered to them. So that's a wrap for this episode. Thank you so much for joining me. I hope you've enjoyed learning about what happens when a B cell meets its respective antigen and the influence of T cells on this process. And I hope you'll join me next time when we cover complement and disease. Have an awesome day, and I will see you again soon for some more high yield learning. Bye.